Good morning, good evening, good night to everybody, and those responsible for organization this virtual forum on TB vaccines. I'm Julio Croda, infectious disease doctor, researcher at Oswaldo Cruz Foundation in Brazil, and professor at Federal University of Mato Grosso do Sul and Yale School of Public Health. I'd like to extend my welcome to all the audience here that watch us in the platform. COVID has been on everyone minding during this conference, a lot of debates, and this panel will discuss in more details what you want to learn with the COVID pandemic and how this can do in the future for TB vaccine research and development. My co-chair for this panel is Gerald Voss from TBVI, and we will moderate this discussion together. You have 6 distinct panelists, and it's very grateful that have this group with us. Let you introduce, you have Nicole, Lurie from the Coalition for Epidemic Preparedness Innovation, Jim Kublin for HVTN, from Hutchinson Cancer Research Center, Eric Anderson from Cape Town HVTN Immunology Laboratory in South Africa, Valerie O'Reilly Mato from Janssen Infectious Disease and Vaccine, Gordon Dugan from Wellcome Sanger Institute, Hassan Mohamed from West Cape Town Department of Health and Stellenbosch University. Let me know, hand over to my co-chair, Gerald Voss, to get the conversation started.
Thank you, Julio, and again, welcome to the audience, and thank you for the panelists to be on this hopefully very exciting conversation that we'll be having that I think cannot be more timely. For those of you who have followed the program so far of the Global Forum, the connection between COVID and TB vaccine has come up basically each and every time, and I think we now have a possibility to examine a little bit further in more detail, and particularly from different perspectives, what the learnings from COVID are actually to advance TB vaccine development. And I think it will be around subjects like the use of platforms, R&D infrastructure, regulatory pathways, but also on the implementation side. And so with that introduction, I would like to get the panel conversation going. And the first subject is a little bit about vaccine platforms. And you all know the prime example is mRNA vaccines, which have basically had their great day and have been developed in less than a year from basically the gene sequence to an efficacy signal. But there are other vaccine technologies as well. And in that respect, I would really like to invite Valerie from Janssen Vaccines to share with us her thoughts on how such platforms are viewed actually from industry, knowing that Janssen in particular has used the adeno 26 platforms in vaccine programs as diverse as HIV, Ebola that has now received authorization, and lately COVID, which is now also authorized for emergency use. So Valerie, how do you see these vaccine platforms and from an industry perspective?
So thank you very much, Gerald, for the invitation and the question. So it's clear that platforms have become really instrumental for vaccine development in the past years. We all went through hard lessons from Ebola and COVID-19, and this has really demonstrated that we need new tools to quickly prevent the spread of infectious diseases. So as Johnson & Johnson, we are a world large healthcare company. So for us, it's natural to actively engage in this area. And it's the reason why we have developed this ADVAC adeno 26 platform. We have shown that we can move quickly to develop vaccines against a variety of infectious diseases. The Ebola vaccine was developed in less than 6 years, and more recently, the COVID-19 vaccine in less than 1 year. But this work comes from many years of efforts, and actually it started really more than 1 decade ago. And for example, it's already the HIV vaccine program that greatly helped us to advance the Ebola vaccine, and then later on the COVID-19 work. And what you have to know is that we also use this platform for other investigational vaccines, such as the RSV vaccine or the Zika vaccine. And all this can happen with the AdVac platform that we use because it enables both the expedited new vaccine development, but also the rapid at-scale manufacturing. We have some generic approach for the manufacturing, testing, and also extrapolation of release, stability, toxicity, and even supply chain profile. And for the development aspect, we accumulate knowledge on the platform data, and that is really supporting the regulatory filing. So that's a little bit the summary I would like to make about the value of platform approach from a company perspective.
Thank you, Valerie. And I think it is a great perspective. And I think what you just said about potentially accumulating data across diseases is that one of the advantages for also basically supporting and sustaining in your portfolio vaccine development for targets that have no commercial background. I'm also thinking about, the Ebola vaccine and for example, data in pregnant women that may potentially help you with COVID as well. Is that the kind of the disease crosstalk that you're looking for as a company?
Yes, we look at our data across platforms, and they are very important. But if I want to go back a little bit to your question, which is how this platform help you to face some challenging diseases like Ebola or others, I think platforms are really important, but engaging in these challenges has multiple routes. So we have invested in this platform for many years to improve it and to accelerate development. So it's clear that it is important, but there are other factors. So for example, engaging in challenging areas has also to do with the company history and culture, and the commitment of Johnson & Johnson in general to try to address major global health challenges. It has also to do with some long-standing commitments to some diseases. So if I take the example of HIV, it's extremely challenging in terms of vaccines, but it's not something coming out of the blue. There has been a full history of investing in HIV and trying to make HIV history in Johnson & Johnson with first 9 different medicines, and now efforts to develop an HIV prophylactic vaccine, which is in phase 2B and in phase 3 in parallel. So it is now tested for efficacy. So this is also an important pillar. And the last pillar that I would like really to mention that is extremely important is that nothing can happen if we do not work with partners. And really, none of these strong commitments would have happened without our partners, and we need these very strong public-private partnerships. So the platforms are extremely important due to the knowledge and the acceleration of development and manufacturing, but they are one element of a puzzle that helps us stepping in these difficult areas.
Okay, thank you, Valerie. This is very thoughtful and insightful to understand, really. And again, there are multiple drivers and multiple things, but I think the platform aspects seem to be very, very important, and probably not only for a multinational vaccine developer like Janssen, but I think across the board for many other entities as well. So maybe with that, I would like to hand over to Julio and to address the next topic with our next panelist. So thank you again, Valerie.
Thanks, Gerald and Valerie. Now you just talk about a lot about the sites and what the sites improve during the COVID-19 trials. The current pandemic has a significant impact around the globe and the conduct of clinical trials for new vaccines. COVID led to substantial investment in clinical trials in different sites. And the HVTN was born as a HIV vaccine trial network, and more recently become involved in TB, and probably with COVID-19 vaccine trials. My question is directed to Jean Kublin, especially to understand what the COVID-19 improved these sites, what you gain with the COVID-19, what the benefits of the COVID-19 clinical trials that do around the world, the different platforms. And he can tell to us what the gain of this investment in the sites for the future, what the next vaccine you can develop in the sites, how you can maintain the sites in the future. Jean, the floor with you.
Thank you so much, Julio and Gerald, for inviting me to the panel. I think this is a great topic and question, and we've put, of course, a lot of thought into this in the past year, as has our primary funder, the NIH and Dr. Fauci at NIAID. And indeed, the foundational work over the past several decades of building the HIV vaccine trials network, where at the beginning of the pandemic, we had over 100 sites around the world who were actively participating in late phase clinical trials, were immediately leveraged to participate in the COVID-19 vaccine effort, and indeed was the foundation for the COVID-19 Prevention Network, or COVPN. And that site infrastructure across the world has really served a tremendous purpose in supporting all of the late phase efficacy trials across these different platforms, namely, the mRNA platforms with Moderna and Pfizer, as well as the two main viral vector programs we've worked with, including the Ad26 with Janssen, as well as AstraZeneca, and then, of course, the more traditional recombinant proteins with Sanofi and Novavax. And we've been very pleased with the effort and the tremendous momentum that has picked up over the past year at these clinical trial sites. Indeed, we started with 100. We expanded that both domestically and internationally to well over 200 clinical trial sites who are participating in these various clinical trials. And we could coordinate that activity centrally here in Seattle, but it was really highly dependent on the tremendous expertise, the infectious disease expertise, clinical trial expertise, and community engagement expertise across the world that had been established with the HVTN infrastructure. So that was immediately used to pivot and leverage against the pandemic with the COVPN. And I'd say the tremendous benefit to the field has been, these results from the late phase clinical trials. We still have a tremendous amount of work to do, of course, and first and foremost among them is interrogating the mechanisms behind these highly protective responses. So I know that's something that Erica will get into, but it is a result of the HVTN's foundational computational and statistical expertise that has been leveraged early on in the very design of these clinical trials, and certainly now being applied to the correlates of protection work that is ongoing. So we've looked at also the lessons learned from COVID-19, of course, that we want to apply to HIV vaccines and TB vaccines in the future. And that spans a number of different topics, including funding, the basic research and development and critical funding of basic research, as well as the support for translational research at our clinical trial sites, the critical community engagement that I've mentioned earlier, and addressing equity very much up front as a need to engage communities and support communities as citizenry science moves forward. So these are just a few topics. I could, I think, expound on it for quite some time.
Great, Dean, for your explanation. And to be more precise, what do you think that in the TB field, during all this investment in infrastructure, what what lesson you can have, and for specifically for TB fielding during the process? What are the most interesting platform and sites that you look in the future for invest in develop a vaccine for TB, especially for TB?
Well, we're very excited to contribute to the TB vaccine portfolios and research and development, and I think that the current pandemic has highlighted the tremendous burden of disease that TB has worldwide, and the syndemics between different infectious diseases, of course, TB and HIV being prominent. And so, you know, we as the scientific community need to partner with advocates and the communities to ensure that the moral obligation that has been repeatedly brought up in the past year of the urgency to develop COVID-19 vaccines is translated to TB vaccines. And I think that is a critical point for us. We've seen a tremendous amount of the pedagogy of science expand over the past year, which has been thrilling. That momentum needs to continue for our engagement, especially of the future generations of investigators, whether it's the early stage investigators who are poised and ready to do research now and find strong mentorship, or even early education in science. You know, these challenges, whether it be HIV, TB, COVID-19, are going to be with us for decades, and there will be new pandemics that emerge in the coming decades, and we need a workforce that is even stronger than we have today. So these are very exciting opportunities for us, I think, moving forward, and each country and the global community needs to continue the momentum of this scientific interest and enthusiasm regarding the tremendous progress we've made in the past year.
Thank you, Jim, for the explanation. I pass to Gerald to make the next question, and you can discuss in the end with more questions that came from the audience.
Thank you, Julio. And I just can't refrain from asking Jim just one more little question. And you know, looking forward over the next years, how would you see this evolve over time? You said there's obviously a lot of your network is now busy with COVID. You know, there is HIV, there's TB, there are other infectious diseases. You know, and I know you don't have a crystal ball, but let's say over the next 5 years, how do you see this evolving, and what are the critical factors for your network to be kind of polyvalent or flexible?
No, thanks, Gerald. Gerald, it's a very good question. So, you know, we've already seen significant changes to our portfolio as a result of the pandemic. You know, we had been trying for over 5 years to get an mRNA platform into the HVTN. Well, now we have several mRNA candidates that are in preparation and will be in the clinic before the end of the year in the HVTN. We hope that similarly we'll see TB candidates on these new platforms that can move into the clinic quickly in first-in-human studies, and anticipate evaluating them whether it be for prevention of infection or prevention of disease endpoints, or therapeutic vaccination strategies among TB patients. So these are direct applications of the emerging technologies in the past year that we're very focused on and very optimistic will bring progress for TB. Similarly, the momentum of the community engagement that we've seen increase in the past year is something we're very committed to continuing across the HIV and TB communities. And all of this, of course, requires funding. And rather than seeing the global community withdraw, given the tremendous successes we've seen in the past year, we really need to work and partner with our advocates and communities to continue to advocate for the tremendous rationale and good sense that it is to invest in these preventive measures in the long term, and how stabilizing that is to communities, to societies, and economies. So there are a number of areas in which we will focus on this. There surely will be significant challenges that we encounter and resistance, but we need to continue to persevere and make sure that this is at a very high priority for the global communities. Thank you.
Thank you, Jim. Yes, for these almost, concluding remarks and the pledge for the future as well. And so, so maybe then I'll turn over to, well, a clinical trial can happen, but it wouldn't make much sense if there were no readouts that would be able to actually measure what is happening in the clinical trial. And for vaccine trials, I think a very important measure is safety and immunogenicity. And Erica, I mean, you are the director of a HVTN supported lab in South Africa. In South Africa, you're in a place where, HIV, TB, COVID, they are all a problem, and Jim said syndemics. I think you're probably in a spot where you're really affected by all those of that. And I'd be curious that from your perspective, you're running such a very important lab, probably also across different diseases. What has been the impact of COVID for you? What have been the biggest obstacles in your experience and the critical success factors for you to contribute to the fight against COVID-19? Erica.
Thanks, Gerald, very much for the question and for the opportunity to contribute to the discussion today. I'll try to answer your question, from our perspective at the laboratory here in Cape Town, but also thinking about the other laboratories from our collaborators, especially here in South Africa. So I think thinking of obstacles, many of the obstacles I think everyone around the world has experienced, but they were particularly difficult when you're trying to do timely research in the laboratory. And so, there are obviously the physical constraints with hard lockdowns, and even here in South Africa, we needed a special permit just to go out to get to the laboratory. Many laboratories around the world have experienced, social distancing constraints on the number of staff who can be there, and have had to adjust, work out shift work, and really have unique ways of motivating staff who can't be in the laboratory normal hours like they would need to be. And then for those laboratories that are working with live virus, the need for BSL-3 space has been, incredibly important, and that's limiting in a normal situation. But in this pandemic situation, it's definitely been difficult, especially for some of our partner labs who are working on antibody work. And then I think one of the other big things has just been obtaining reagents, supplies, and critical controls in order to develop these assays. So as a laboratory in a developing country, we're used to having long lead times and obtaining reagents due to, imports and exports and, third-party vendors, et cetera. And now all of a sudden laboratories around the world have also been experiencing this. So everything from PPE for laboratory staff to, custom antibody conjugates for flow cytometry, and our colleagues in the molecular space have had huge competition with diagnostic labs for instance, PCR reagents. So that, that's definitely been a challenge and a barrier to doing things as fast as we would hope to be doing them. The other thing that was really critical was to get specimens from COVID-infected individuals. And being part of the HVTN network, we've been very fortunate that there was early access to different cohorts of infected individuals. But, for people who are starting this anew, they had to find new clinical collaborators, write protocols, get ethics approval, and cohorts enrolled really quickly in order to just have the specimens to get the assays up and running in their laboratories. And they needed enough of different types of specimens, different specimens from people in different stages of illness to really work on these new assays. And the other obstacles have been, funding. So many laboratories were able to pivot quickly, but needed funding to get up and running. And so I think, getting the funding there to be able to acquire the supplies, reagents, et cetera, that were needed was really critical. In terms of the successes, I think, there's been huge advances in assay development to measure immune responses to SARS-CoV-2. Many labs were able to pivot very quickly, leveraging their existing assays, and especially in our clinical assay environment where we have validated assays and we have a very good knowledge of the different parameters that affect the assays, we could pivot quite quickly to making sure that we could validate new reagents and new aspects of the assays to apply them to SARS-CoV-2. There's also, really fast and extensive sharing of both reagents and information. And information has really been touched on in this conference already. In terms of the reagents and samples, there were, many, many new collaborations that were formed. People begged and borrowed, but tried not to steal reagents and consumables from other laboratories to get new assays up and running. New collaborations between labs and clinics, even within institutions, people who weren't working together necessarily, all of a sudden form new collaborations that will really endure beyond COVID and will help us apply, the lessons of COVID to TB and to other pathogens. And new consortia were formed, for instance, the South African Variant Consortium. One of my colleagues has said that she actually has 20 more collaborators than she did 6 months ago. So it's really, catalyzed people working together, sharing data, sharing information, and everything from high level down to the small details that would actually save lab scientists months and months if really details of somebody else's protocol of how they're doing something. So people have been very open. There's been extreme goodwill, I think, among researchers to really share information. And then as Willem mentioned, and others have in his talk yesterday, there have been unprecedented levels of data sharing. So high use of preprints, open access to COVID publications. The other thing I wanted to highlight is the virtual meetings that have occurred this year have really allowed people from throughout the world to exchange information, to learn from other scientists, and people who maybe in the past wouldn't have been able to participate because they couldn't travel and it was a budget constraint, all of a sudden have access to all of the cutting edge data. And so that's been really critical. And I think everybody's really woken up to the fact that the pace of research is too slow, and we need to apply this accelerated year, which has, driven some people to the breaking point probably, but we've got to keep this, as Jim said, keep this momentum going and really find ways that we can evaluate what we're doing and really speed it up, because there are ways to accelerate what we're doing. And one of the biggest pieces of that, I think, is the sharing of information. As Jim also mentioned, there's the development of young investigators. So there's been great opportunities to publish new assays, new findings about a totally new pathogen. And there also seem to have been a lot of interesting opportunities for sort of early to mid-career investigators to take on more leadership roles in consortia as in other situations, because their PIs are totally overwhelmed and overworked. And so it's been easier for people to pass on, okay, you're in charge of this, you run with it. And I think, a lot of people are finding that they've been able to really step up to the plate and take leadership roles in some of these situations. And I think that's been wonderful. And I think the other thing is just really, a new pathogen, new interactions with the human body. This has really reignited people's interest in science, and hopefully they can now translate that back to the other pathogens that they were working on before this all started. So it's been, it's been a time of trials and tribulations, but I think also just amazing successes. And I think all of us feel so grateful to have been, doing this in this time, because it's such a critical and fascinating sort of revolution in science right now.
Right. Erica, thank you for this very engaged and remarks, and I'm really the last one also about the opportunities, in particular for young investigators, is something that we should keep in mind. And so I think a crisis also comes with opportunities, and I think that is a really good example. And the other one, which I think you rightly said, so is also data sharing and collaboration. What an experience, and I hope that is something that will be maintained even after the pandemic. This is great. Thank you really for your perspective, which is really great. And before handing back to Julio, I just wanted to alert our panelists to the fact that in the Q&A, if you can see it, there are a number of questions, some to specific panelists. And if you have an opportunity to answer to those questions as we go, or if the staff behind the scene can make the Q&A visible to the panelists, then that might be helpful. And we will try to address, some of these questions if we have time at the end. So just a little logistics remarks, and then I hand it over to Julio. Thank you.
Thanks, Gerald. And you talk a little about funding, and this, I think this is the quick question now. Nowadays, you have new platforms, new clinical sites that have infrastructure, new labs to perform these studies in TB vaccines, and you have a new the next session. It's talk about this, but you have opportunity now in this section to discuss a little bit about this research and development of vaccines like malaria, HIV, and TB, or other disease needed this funding. And that Wellcome Trust has been an incredibly important contribution. Scientific advance from this investment, it's helped a lot to accelerate the response to COVID-19. And I'd like to put Gordon in this conversation, and he can explain to us what the space for TB vaccine, what you can do relate to TB vaccine, and what you learn with the COVID-19 vaccines, what you can do for move very fast and try to fill this gap in the investment in the TB vaccine to be more rapid response to the TB. And I think you have a good moment now to discuss this, because you just almost clinical trials and the majority of the world almost run or finish, and you can join out this effort for another disease. And Gordon, I want to like to hear from you how you can improve the funding for develop a new TB vaccine, and what you can learn with COVID-19 vaccines.
Yeah, so obviously the Wellcome's been thinking about infectious disease, particularly epidemics, for a long time, and we were just reorganizing our strategy just as the COVID epidemic struck. And we've decided we're going to keep our broad scientific platforms going for discovery, but we're going to focus on 3 areas for impact and translation. One area is infection, which you'll be pleased to know, which I'm looking after at the moment. The other area is climate change, and the other area is mental health, which I think are all 3 relevant to what we've seen occurring during the last year. And that will allow us to actually focus our funding. So rather than trying to fund everything, we're really trying to fund in a more targeted way. And we have had some preparation for tackling disease. We were involved in the formation of CEPI, which has been focused around epidemics, but we are now more interested in diseases that have the potential to escape therapy, for example, AMR, and TB is a great example of that. And another thing we were investing in, we invested in the first TB genome. We funded it between the Institute Pasteur and Sanger. And actually our genomics funding has proved to be very valuable, I think, during the COVID pandemic. We funded COG UK, where we sequenced 500,000 genomes and allowed us to identify variants by providing this as open access data. It's allowed us to provide information at a global level and through GISAID and what have you, which has allowed, and it allows investigators working in remote areas, in low and middle income countries, first of all, to access the data, as we just heard previously, but also informatics and data management is accessible to all if you've got the right infrastructure. The other thing I've been involved in personally is that the regulatory authority I've been involved in as an expert advisor for the MHRA regulators in the UK. And it's been a real roller coaster of ripping up the old regulatory form book and reinventing it. And what's happened is the pace and change has been tremendous. Whereas previously you might have done animal studies, people are much more interested in human studies. So one thing we're thinking and will be funding is human challenge and human investigations. They can be around in individuals in low and middle income countries as well. We've just funded the first COVID challenge studies. They're ongoing at the moment. I've seen the first data from the volunteers. It's very interesting. And I think that's very important. But the other thing we're going to roll out is a global surveillance network, which will be built around genomics. And we're going to do it by investing in young investigators in low and middle income countries. And some of those sites will be where TB is prevalent, where HIV is prevalent, where dengue is prevalent, and where COVID is now prevalent. So the aim there is to create sustainable hubs over a long period of time. We're going to drive it through using genomic surveillance, but then we'll build on top of that secondary phenotyping. Might be the diagnostic test we just heard about. It might be the testing of drugs and antibiotics. And then we come to TB and TB vaccines. So one thing we are thinking about is whether or not to invest in a large-scale program on TB vaccination. So the way we would probably do that is we would maybe back one or two vaccine candidates, but we're also interested in innovation, and some of the speakers have talked about mRNA vaccines, but those have to be affordable. We have a small entity called the Hillman Laboratories, which explores how to make affordable vaccines for everybody. We will also invest in low and middle income companies in a targeted way in vaccine manufacturing capacity. We've already identified the areas we're going to work in. And if TB vaccines come along, they would be one of the areas that we would be supporting. So we're going to do that again over a sustainable period. So in summary, really, I think what we want to do is use our, if you like, our funding power in a more targeted way. It's science-driven, it's investigator-driven, it's going to be globally driven, and TB is on the agenda.
Great, Gordon. I think it's very nice for your audience to listen this for you. It's in this forum. It's very nice, and you have this during the last year, this concern about the gap about the funding. Do you think you can able to convince the government, all the founders, the philanthropic government, how we can try to convince the majority of the people that founded this kind of projects for RNA vaccines, adenovirus vaccine, to put more money in this specific area? How you can deal with this? How the best argument to involve the stakeholders?
Yeah, so I think I'll repeat myself a little bit. I'll say our focus is scientific excellence. And that scientific excellence can be applied to the production of medicines. But if it's going to be to the production of medicines, it has to be accessible medicines, okay? And we're looking, for example, at vaccines, but we've also just done a big survey on affordable antibodies. So can we actually realistically bring antibodies into view as well, and small molecules or what have you? And so we're definitely going to be doing that. And we are influencers. And so during the pandemic, the British government listened to us, and they backed genomics, which they probably wouldn't have done, right? And so it meant that COG UK, we were able to get get the money invested to sequence large amounts of genomes without any guarantee that we would get payback. And now they're seeing the payback in the form of variant identification. So they're now believers. So what we have to do now is to lead by example. And so what we're going to do is invest into particular hubs in low middle income countries and try to use those as spikes through young investigators, but to do it over a long period of time. We cannot just do it for a year. We cannot do it for 2 years. And we're thinking about mechanisms of funding at 2 levels. One is a mechanism of funding whereby we can keep sustainable funding. But the other thing is about the cost of reagents and equipment. So I was asked to provide MySeqs for Bangladesh about 4 years ago, and I was quoted $100,000 a machine, $200,000 machine. So I went to the procurement office at the Sanger Institute and said, can you get me a discount? And they got me a 90% discount.
Great.
And that shows you the power of working together as a community. And the previous speaker, I think it was Eric, has mentioned about the cost of reagents, etc., in those settings. We have to focus on all levels to enable the investigator in low and middle income settings to be competitive and to be on an equal playing field.
Thank you very much, Gordon. The time is urgent. I pass to Gerald for the another topic, and to another panelist can have opportunity to discuss another aspect, but it's great you can talk more in the end of the panel about this funding, and you have another section after that discusses the same issue. Gerald,
thank you very much for inviting me. Thank you very much for inviting me. It's a pleasure to be here.
Thank you, Gordon. Thank you. And Julio, as you said, I'm conscious of time, and we are running a little bit late, at least if we want to have a little bit of a wrap up at the end, it might become tight. So without further ado, let me then turn to Hassan, just by saying that these COVID vaccines have been developed in record time. But then what about implementation? And we felt that the implementation side of things is obviously equally important for these new vaccines. And what are the challenges in implementing COVID-19 vaccines, in particular in populations where there are no well-established delivery mechanisms like the EPI in infants and children? So Hassan, from your perspective and experience in South Africa, how has that been? And then how can that help future implementation of TB vaccines? Thank you. Over to you.
Yeah, thanks so much, Gerald, and thank you very much for this opportunity to participate and to connect with people that I've previously worked with. I've scanned the participant list, and it's nice to see some friendly faces amongst the participants. So coming specifically to your question, and of course in South Africa we do have an established EPI program, but of course the COVID vaccine is targeted at a different group, and it's targeted at adults and the elderly. And of course this has posed many challenges. But I want to start off by saying trial design matters. And in order, the first step in implementation is, what does the science say? And what did the trial results show? And I think it's absolutely important to get clear signals from the registration trials so that it enables policymakers to implement easily. Both efficacy and safety signals need to be clear. And I think our experience in South Africa has been mixed. Different vaccines have had different kinds of trial results, and the trial designs, from my point of view, have influenced the way policymakers have responded. So we need to think ahead. We need to think about research translation. And I think it's, as scientists, as clinical trialists, you need to think ahead to the implementation phase when you design your clinical trials. Secondly, of course, a massive issue which is global is vaccine hesitancy. And of course, with vaccine hesitancy, in fact, has been around since, the time of Edward Jenner and the first smallpox vaccines. So it's there, it's in our nature to be suspicious of anything new. And likewise with COVID, that was there. But of course, in addition, we've had the challenge of misinformation and social media that has facilitated and influenced vaccine hesitancy. So vaccine hesitancy has become a massive challenge. We cannot ignore it. We need to address it, but we need to think very carefully about how we address it. And for me, it's not about engaging anti-vaxxers. It's really about promoting the positive narrative around vaccines. And it is possible. And my experience in being involved in implementation shows we can make a difference. We can address vaccine hesitancy. We can deal appropriately with vaccine information, but we need to think about how we do it. We need to plan for it. We need to expect it. And with social media and the rapid global spread of misinformation, and South Africa gets most of its misinformation from abroad. It doesn't originate in South Africa, but it just takes a click for that information to spread virally around the world. So I want to give an example, and that is, for example, even amongst health workers, when we surveyed them about taking a COVID vaccine, just over 50% said definitively yes, that they would take a COVID vaccine. 20% said definitely not. And there was about between 20 and 30% that were unsure. And these are the people who are meant to be administering these vaccines in the future. So quite a high level of hesitancy at the very beginning. But through information sessions, through engagement, and talking to people, and once vaccinations actually started, and this is the global experience, more and more people were willing to be vaccinated. And our subsequent survey results show that more than 80% of people were willing to be vaccinated. But again, it's an example of how important vaccine hesitancy is. Cold chain matters, and of course we know that different vaccines have different cold chain requirements, but of course these are not insurmountable. We are getting the Pfizer vaccine, which we know has quite strict cold chain requirements, but we've made a plan. We've made it possible, and in fact through research infrastructure, through refrigeration capacity at one of our universities, they're actually assisting us with storing the Pfizer vaccine. So in fact, there's been a partnership, mutual benefit that we've gained through research capacity that's assisting us with our vaccine rollout. The next issue I want to deal with is the whole question of sequencing. And as you know, we have vaccine shortages. There's a limited supply. And of course, this is an issue that's going to become a problem if we develop a successful new TB vaccine. The demand will outstrip the supply, and it'll take time to reach all communities. So the whole concept of sequencing, who gets it first? And even if you say, yes, health workers should get the vaccine first, who amongst the health workers should get it first? And if you say, well, patient-facing health workers, and then the next question is, which patient-facing health workers get it first? And we had this interesting concept of vaccine envy, where you have health workers who are unhappy because they haven't received the vaccine and feel that they should have got it ahead of someone else. So it's an interesting phenomenon that we've observed, with the rollout of COVID vaccine, because in a country like South Africa, it's been a trickle. We haven't had the resources that first world countries have had to get large volumes of vaccines. So it's been a major issue for us. So I'm going to stop there. I mean, I think that's a range of important operational implementation issues that we in the vaccine world we need to think about as we move ahead with TB vaccine development. Thank you.
Thank you, Hassan. And I couldn't agree more that just I think a very, very pertinent reminder of this, end-to-end perspective that we need to adopt from discovery right to implementation, with its challenges and opportunities basically at every layer and every step. So thank you for this insightful thoughts. And I would like to hand back to Julio for introduction of our last panelist. And we are right about on time still. So Julio, please, and thank you.
Thank you, Gerald. You discuss a little bit now about how you can prepare for the next pandemic, how you can deal with this. The world has united behind the development of COVID vaccine in unprecedented manner, show that vaccine can be developed in less than one year to COVID-19, and CEP has made major contribution to this success. And continue to think about the preparation for next pandemic. And this is a key for us. In the same time, you have some hope, especially to other infectious disease, and now especially for TB, that continue to be a huge burden in global health and pose a future challenge in the antimicrobial resistant also. And my question to Nicole, it's how to use COVID-19 to make the case for investment in another global infectious disease. How you can look for future epidemics and pandemics, how you can accelerate this responsibility after the COVID-19. Nicole.
Yeah, well, thanks for the question, and thanks to the other panelists too for incredibly insightful and provocative, and also just really optimistic comments during this time that we're all facing. You know, I think the first thing that I would really note is and others have, is that the success of COVID has built on years and years and years of work, whether it's in vaccine development. We all talk about, gosh, it's a miracle we developed a vaccine in under a year. We didn't do that. We've been working on these platforms for 10 years. And so fortunately, a lot of them have been ready to pivot to a new pathogen. We've been working on HIV vaccines since, I began training in medicine, just about. And and same with TB, and we've made enormous progress along the way. And so I feel like that there are a couple of pieces that we need to think about going forward. We have these amazing centers of excellence around the world. Many of them are pathogen focused, and there are these research communities that focus on specific pathogens. And I've come to learn that many of those research communities each have their own personalities. There are these research communities, fortunately like yours, that also focus on syndemics. And so you've actually been able to see a lot of the synergy that goes from one to another. But even when COVID first showed up, one of the things that I was really struck with is that while lots and lots of these pockets and research communities sprung into action, and they've done really amazing things for some really key areas, it wasn't really clear, like, whose role and responsibility is this in the world to do something specific? So whose role and responsibility is it to get and share and grow up virus around the world? As Erica talked about, whose role and responsibility is it to collect samples from patients in different levels of illness and share, et cetera? And so going forward and thinking about what does our preparedness system need to look like going forward, I think in the past I've likened some of this to a conductorless orchestra, and you've got a terrific string section and a terrific brass section and terrific percussion. But we need, I think, a little bit more integration there. So as we think about going forward, and I think about the preparedness investments now from CEPI, and as CEPI is looking at 2.0 version of CEPI and saying, well, what do we need to do differently? There are a number of things I think that come to mind. One for us is that it was great to develop this vaccine in under a year, but an awful lot of people died in under that year. And that's way too long. So we now have sort of a real moonshot goal aspiration to figure out how we're going to do this in 100 days. So if we figure out how we're going to do this in 100 days, there are all of these parts of the system that have to be tuned and ready and pre-deployed. So we heard from Jim about this amazing set of research networks around the globe that we're able to spring into action. And the other thing that we need to remember is those research networks need to be where the disease is if you're going to do this, and it's sometimes hard to predict that. So how do they stay warm? How do they stay pre-positioned? Erica told us we brought all these new scientists into this. We have to be sure there's work for them to do now in this inter-epidemic period, right? And so how is it that we think about these different pieces of what we're going to develop and keeping warm? Gordon appropriately and others have talked about how expensive the mRNA vaccines are. Well, shouldn't our challenge now be to use science to improve those platforms and make them easy to make a vaccine from, easy to manufacture, reduce the cost of goods, and make them as accessible, just as an example, make them accessible in different ways around the world. So one part of CEPI is focused on now, what is the moonshot and what do we need to do for 100 days? A second part is being sure that we are true to our equitable access mission and connect particularly in less advantaged regions around the world. And so the idea that Gordon's put out of having these science hubs that can spring into action around the world and with a focus on equity, very much a focus here. And if we can get out of the disease pathogen model and think about the pluripotential capacity of these disease hubs, I think the TB world and others are going to be very well served. And, you know, TB is going to be with us across all these epidemics. So it is a great sort of poster child, along with some of the other diseases we know about, for work that has to be done in the inter-epidemic period. We've just signed an MOU with the African Union that's looking at broadly at manufacturing capacity in the African continent. And CEPI's contribution is mostly going to be manufacturing innovation and how to make manufacturing faster and easier and less expensive. But that has to situate and coexist with clinical trial capacity and clinical trial networks and laboratory capacity and sequencing capacity and community engagement capacity and all of those pieces, so that the preparedness for the next pandemic builds on what we've done here, but it also has a day-to-day function, keeping people busy doing really, really important work, continuing training, continuing to advance the science in all of those things. There are a couple of areas that I think we need to work on well before that. And I was interested in Jim's comments about the trial network, and maybe just to stimulate a little bit of conversation now for the next part of our discussion. One of the things that came out of the Ebola outbreaks in particular were some really innovative clinical trial designs, particularly in the therapeutics. And we got to the point where we could do platform trials and therapeutics and adaptive trials. And we saw those be used for therapeutics, particularly through the recovery trial and through solidarity. We haven't done that in vaccines. We've seen one trial at a time, each with having to recruit 40,000 subjects, half of whom are in a control group. We don't have a really great sense about how different vaccines compare to one another, et cetera. I think as we start to move forward, that's an area that's ripe for innovation. It's one of those things that needs to be pre-positioned. We did this in record time, but again, if those kinds of concepts and things were pre-positioned along with the deployable pre-funded research capacity, we'll be in much better shape. And then finally, if this infrastructure is warm and funded, it still needs a huge amount of funding when it needs to spring into action. For vaccines, we heard there was no money in the world to buy raw materials for vaccines, to make vaccines, to guarantee their purchase, to do all those things. So superimposed on all of this that we're talking about has to be a global financing system so that we're not passing the tin cup in the middle of the next pandemic. So thanks.
Thank you, Nicole. You have a short time to end the section, but section is very encouraging for the entire scientific community. Listen, these key persons in this section, leaders for us some hope for the future, especially for the TB community. And you're very hopeful to develop a vaccine for TB in the future. And I pass Gerald to make the closing remarks for this section. and you can end the section if you have time for 1 or 2 questions, but Gerald decide, depend of the time.
Yeah, thank you, Julio, and also thank you to all the panelists. I feel like we could continue this discussion for hours. Basically, we only have scratched the surface on so many different things, but what I did perceive throughout the different topics and contribution is really optimism and opportunity, and rising from the pandemic with a new perspective. And I think overall, not only for vaccines, but in particular vaccines, this is a great moment in time. And what has happened during the pandemic, I think, has changed vaccine development and vaccine implementation for the future and across different diseases. And so I think we are really hopefully be able to translate our learnings from the pandemic into the future more broadly across infectious diseases, including TB. And with that, I think we are really now at time. I would again like to thank all the panelists, my co-chair, and the audience for this really engaging and inspiring discussion. And I would like to close this particular panel. Thank you.